Good evening, everyone. So, my name is Jyoti Sharma, and this is week 8. And this live session will be from 7 to 9. So, can you please confirm whether I'm audible to you or not?
Yes, ma'am. Good evening, ma'am.
Good evening. Hello. Okay. This is what we have learned. We started from primary treatment, then we went to secondary treatment, and now this week we will talk about tertiary treatment, or we can also say like advanced treatment. uh, just let me give you little brief that in primary treatment we talked about different bar racks, bar screens, then grid chamber, equalization tank, sedimentation tank, and we also uh, solve some numerical based on some techniques, how what kind of dimension we should have of that particular tank and all. So this was under primary treatment equipment, in which some to some extent we can remove the BOD uh, and the minerals and nutrients from your wastewater. Then we went to secondary treatment, and in which we also studied how we can use lagoons, aerobic digestion, activated sludge process, anaerobic digestion, UASB, biofilter. We talked about this, right? So these were the part of the secondary treatment. And within secondary treatment, we also had some advanced treatment. For example, granule activated sludge process, sequential batch reactor, membrane bioreactor, phycoremediation and phytoremediation. So till phytoremediation, we already have studied in the last week. So in this week, what we will cover, these two, electrochemical system and microbial electrochemical cells. So this we will discuss in this week. Plus, we also discuss tertiary treatment. So this is actually what we will discuss today. So in tertiary treatment, we have three types. The one is like using adsorption. We can have the filtration, then using membrane processes, and using different advanced oxidation processes. So along with this secondary treatment, we will try to cover these three, and within that we have like separate at length uh, MCQs on membrane processes, and we will talk about different type of membrane that we can use and what different size we have for that particular membrane. Then what kind of shape we have in this, and even how the wastewater will transport against this membrane. Okay, so we will talk about this, and then we also touch advanced oxidation processes. This is very important because right now whenever we are using reverse osmosis or RO in your household, so we also use some advanced oxidation processes. For example, we use ultraviolet radiation along with ROs to treat the water. So this is what we will cover today. So let's start with secondary treatment process. Again, I have a lot of MCQs question, and you people are welcome and even encouraged that you please do the participation, give me the answer, and don't worry about the wrong answer as well, because we will learn together. Okay, so this is what we will cover from starting secondary treatment processes, and then we will go to the tertiary treatment processes. Okay, so my first question is on microbial electrochemical cell. Which component directly act as the biocatalyst for converting organic matter into electricity or hydrogen? And we have four options here: microorganism floating in water, biofilm formed on the electrodes, electron carrier in the cell, and then treated wastewater. So what do you think? What is the right answer for this? From that terminology only, you can tell me the answer. If it is a microbial, then electrochemical cell. So from that, I think you can get the answer. Okay, so let me see who gave me this answer. Uh, Venkateshwara gave me B answer. Yes, I also agree with this B. So because we right now we are talking about this secondary treatment, and secondary treatment we people use microorganism in the secondary treatment. So obviously if we are talking about this microbial electrochemical cells, then we will use the microorganism. The first thing is very clear. The second thing is, if it is the electrochemical cell, so it means we will have anode, we will have the cathode, and this will be attached to some voltage, may or may not be attached to some voltage, and after that there will be some charge that will flow from anode to cathode, and this is how electricity will produce. Okay, so the thing is, here if we are using microorganism, it means, whenever there is any wastewater, so let's for example, if we do have any wastewater, waste water will have some organic matter, and here will the role of this microbes. These microbes will do the oxidation of this organic material, and this is how this will participate in electrochemical cycle. And this is how, just using the wastewater, just using the organic matter that we have in the wastewater, we can generate the electricity. So on the electrodes we have anode and cathode. So mostly on anode we do have the biofilm of microbes. Bio is just the microorganism, so we just have this microorganism are attached to the anode, and this is how it will act like the biocatalyst. So that's why this B answer is here, right one. This is just the brief introduction, because we are talking about bioelectrochemical. So till now in your like up to 12th standard, you have read about this electrochemical system. So this is the device that can generate electrical energy from chemical reactions. And in bioelectrochemical system, microorganism will help for this chemical reaction, right? So this is something we are here. We do have energy rich wastewater, and there we will have some microbial metabolism, and we do have a lot of things. We will read about this, don't worry about this. And then we will have the electricity, hydrogen, and some carbon, so such kind of things will be produced. So this is something about bioelectrochemical system. So here also the definition is here. So this system convert biodegradable organic matter to electrical energy or hydrogen using a biofilm on electrode as a biocatalyst. So this is very simple. We are having the chemical reaction to generate the energy, but here along with that we are using microorganism. So this is how it will work. And we do have different type of system, bioelectrochemical system, based on our need. So for example, if we need energy, so we can use this microbial fuel cell. If we want to do something else, for example, if we want to do electrolysis, then we will use microbial electrolysis cell. So these are different type of your BES, that is bioelectrochemical system. Fuel cell, electrolysis cell, electrosynthesis cell, enzymatic fuel cell, microbial solar cell, plant microbial fuel cell, and microbial desalination cell. So these are the application, extended application of your bioelectrochemical systems. And here in this right now in this live session, we will talk about this one only, microbial fuel cell, right? So my next question mostly will be on microbial fuel cell. This is just the illustration that how we can use different cell, that is the part of your bioelectrochemical cells only. So this is just the illustration. Okay, so my next question is, which of the following is not a typical product or output of microbial electrochemical system? And we have four options here: electricity and hydrogen, methane and alcohol, treated water, ex vivo protein complexes. You people are encouraged to give me the answer, and don't worry about the wrong answer because we will discuss, we will learn together. Okay, so Venkateshwara gave me D. Yes, obviously. So this is the part of your process. This is not the output. This is something that we need for the chemical process, for the chemical oxidation. And this is not the output. Output will be treated water. It may be methane or alcohol. And obviously, if it is electrochemical, that means we are using chemical to generate the electricity, then electricity will also be generated and along with the hydrogen. So these will be three products that would be produced by your microbial electrochemical system, but not ex vivo protein complexes. So yes, D is the right answer. So this figure is once again showing you, just see at the bottom what we are getting: electricity, hydrogen, then methane, alcohol, and treated water. So this is what we will get. And this ex vivo protein complexes are the part of MXC. So that is microbial electrochemical cell. So these are the part, but not the product. Okay, so as I told you, because it is electrochemical cell, right? So we need anode and we need cathode as well. So you have to tell me the combination is least likely to present a correct pairing of item and material in microbial fuel cell. Anode, graphite felt; cathode, reticulated vitreous carbon; protein exchange system, sulfonated polystyrene; electrode catalyst, carbon cloth. So this is something, if we have some item, for example, if it is an anode or if it is a cathode or if it is a electrode catalyst, so that should be made up of something, right? And you have to tell me the correct pairing. Okay, so this is least likely, so which is not the correct pairing, right? Venkateshwara, you need to change the option because B is not the right option. So B is the correct pairing, right? B is the correct pairing. But in the question, I asked you which one is the least likely to represent a correct pairing. So yes, so Dia gave me D, and yes, D is the right answer. Let me clear this and let me show you the table. So but before that you have to learn what is this microbial fuel cell, right? And if I'm talking about the material, then like what item we have in this microbial fuel cell. So as I told you, we have anode, we have cathode, and to the anode the attach is your microorganism. So this is your bacteria. So here we will have the oxidation. At the anode we do have oxidation, and this oxidation will lose the electrons, so can you see these electrons? So this will transfer electron from anode to cathode, and this is how it will produce water and all. So just see here, this microbial fuel cell harness electrical current from microbial oxidation of organic matter using a solid electrode as the electron acceptor. So whenever there will be microbial oxidation of organic matter, so this electron will be taken up by anode and then it will be transferred to the cathode, and this is how current will be move on and the electricity will be generated. The anode surface facilitate microbial attachment, and oxidation of organics, therefore generating electrons that are which are then simultaneously transferred to the cathode compartment, and this is through the external circuit, and this is how electricity will be generated. So this is about the microbial fuel cell, but my question was about anode, cathode, and cathode catalyst, right? So let me show you. And this is different organism. So here it was written bacteria. So what kind of bacteria? You should also specify the name of that bacteria, right? So that most of the time the bacteria is rhodophyrex, and we also use geobacter. So these are the direct uh, so they are used for the direct oxidative metabolism, but along with that we can also use Escherichia coli and all, right? So but these two are very important. Now I'm coming this I already have shown you that this is your organic matter, and then using the microorganism we will have some proton and we will have the electron. This electron will be transferred to the anode and to the cathode, right? And this is how it will move and it will generate the energy, and this is how it will also treat your wastewater. Coming to the answer of your MCQ, so for the anode, the material may be graphite felt, carbon paper, carbon cloth, palladium, black and reticulated vitreous carbon. But here, if I go to the question, this one, for the electrode catalyst, let me show you. For electrode catalyst, we have polyaniline electron mediator immobilized on anode, palladium or platinum. I don't know about this, but you can read about this. Then magnesium oxide and ferrous. So these are the things that we have for the electrode catalyst. Okay, so among that, so this was not the right, because along with that it was carbon cloth. So carbon cloth is not the right option for your electrode catalyst. So that's how D answer was the right one. Okay, so coming to my next question, in that electrochemical cell we can use pure bacterial culture. It means we will have very specified species of the bacteria, but we can also use mixed bacterial culture. It means we can have different type of bacteria within that cell. Okay, so the question is, which of the following is not a limitation of using a pure bacterial culture in microbial fuel cell? And we have four options here: high substrate specificity, slow growth rate, high risk of contamination, higher power output. And the question is not a limitation, right? What do you think? What is the right answer? Am I get the right answer? Okay, let me see. Okay, no one is giving me answer, but okay. Let me tell you, D answer will be the right, because we are talking about the advantages of pure bacterial culture. We are not talking about the disadvantage, because this is not a limitation. So we are talking about the advantage, right? Uh, then higher substrate specificity. Uh, if we need very particular substrate, for example, in the wastewater, we should have very particular organic matter in it. So this would be the disadvantage, because wastewater may have a lot of variety of organic matter, right? So if we do have pure cultures, then pure culture may need very specific substrate. very specific food to eat, right? And if we do not have that food in our wastewater, that means that pure bacterial culture will not work. So this is disadvantage. This is the disadvantage of your pure bacterial culture, right? Slow growth rate, this is again the disadvantage. High risk of contamination, because it is pure bacterial culture, then there is a higher possibility of higher risk of contamination. So this is again uh, a disadvantage, right? But the advantage is it can produce higher power output. So that's how D answer is the right one. And here also you can see for the pure culture, that may show high electron transfer efficiency, but it have very slow growth rate, very high substrate specificity. Mostly it need acetate or lactate, and relatively low energy transfer efficiency compared to mixed culture. And if we are talking about this mixed culture, then that obviously if we do have mixed culture, it means we will have a lot of biodiversity within that bioelectrochemical cell. It means it will have higher resistance against process disturbances. uh, it can take a lot of substrate, so there is no that specificity for that substrate, right? Then obviously then a smaller substrate specificity, but the consumption rate will be higher because we do have variety of microorganisms, and it will give you the high power output. So this is how these are the advantages and disadvantages of whether we are having the pure bacterial culture or mixed bacterial culture within our uh, bioelectrochemical cells. Okay, so I think based on that explanation, you can tell me the right answer for this. And this is compared to pure culture, mixed bacterial culture in MFC are characterized by, and this is low substrate consumption rate, but high electron transfer efficiency, high resistance to disturbance, and broader substrate utilization, greater risk of contamination but lower power output, and fourth is faster growth rate but higher substrate specificity. So what are the characteristics of mixed bacterial culture that we will use in microbial fuel cell? Okay, so let me see the answers. Okay, so Venkateshwara gave me B. Yes, so we already have talked about this, right? If we do have biodiversity, a large biodiversity, then there will be higher resistance to the disturbance. This is something like in the forest, when we do have different type of plant species or tree species, then whatever disturbances we have due to, let's say, climate or due to fire, but that disturbances will be resisted because we do have a lot of plants or trees. uh, the loss in one tree can be compensated by the benefit from another tree, and this is how it will balance that disturbances, right? And this is how what is happening in the mixed bacterial culture. If we do have biodiversity of bacterial culture, then obviously there will be higher resistance to the disturbance and more substrate utilization, broader substrate utilization will be happen. This is something like this. If we do have different eaters, different type of eaters in the food chain, then it will be known as food web, right? Because the first tropic level will be connected to the tertiary, secondary, and with the herbivore, and we do have different type of microorganism or organism at each the places. So it means the one will have a lot of option to eat. Okay, so for example, if it is a deer, then deer will have, let's say we do have different variety of grasses, then obviously it will be the broader substrate utilization, so something like this. So this is the advantage of mixed bacterial culture. So that's how B is the right answer. Okay, so the next question is, which of the following is least likely to directly influence microbial fuel cell operation? And again, we have four options here: electrode material, material, type and composition of substrate, operating condition in anodic chamber, shape of the external power circuit. The question is least likely, so should not be the correct one that will directly influence microbial fuel cell. So first you have to list out what are different factors that would influence the microbial fuel cell, and then what is that one that would not influence the microbial fuel cell. Okay, so Dia gave me the answer. Yes, I also agree with this one. So the shape of your external power circuit is, question is not visible, is it? Uh, it may be due to internet problem from your side, I think, Venkateshwara, because Dia is able to give me the answer. So it might be that you have to disconnect and connect it again. Okay, so yes, let me show you different factors on which your microbial fuel cell will depend. So the first is electrode material, what kind of material you are using, either it is carbon cloth or different type of material that you are using, what pH that you have of that buffer. So you have anode, you have cathode, and these are dipped in some buffer, right? So what is the pH of that buffer, and even the electrolyte, then protein exchange system, then operating condition in anodic chamber. So this is anodic chamber and this is your cathodic chamber. So what operating condition we have in anodic chamber and what operating conditions we have in the cathodic chamber. Then type and composition of substrate, as I told you, based on that also you will select what different kind of microorganism that you will use in that bioelectro chemical cell, right? So based on that also your operation will be dependent on. Then type of oxidant used in cathodic chamber, and presence and absence of catalyst, because that will accelerate your process, right? So these are different factor on which your operation of microbial fuel cell will be dependent on, but not upon the size of shape of the circuit, external circuit, right? So that's how the answer was your right answer. Okay, because there is electricity, right? Then there is a electricity current, so current is by I, and then we also have to give some voltage. So or so this is V, and you have to tell me in the microbial fuel cell the relationship between cell voltage, current, and external resistance is given by V is equal to IR, V is equal to I by R, V is equal to R by I, and V is equal to I square R. Okay, so okay, I got answer, Venkateshwara and Tia gave me A answer. So obviously, we people are learning this Ohm law since school time, and this is just based on your Ohm's law, and this is equal to V is equal to IR. And let me show you. So that's how A is your right answer. And so this is your, whenever you will have any electrical circuit, there you will have some voltage and you will have some resistance as well, and there is a relationship between V R I, and this is this is I is equal to V upon R. So this means V is equal to IR, right? Okay, but in your microbial fuel cell, we have different terminologies or efficiencies or the losses. Based on that, you have to tell me the cell voltage that can be expressed as, and we have four options here: A, B, C, D. So what will be the right option? I will discuss about it in next slide. Okay, Dia and Venkatesha both gave me a B answer. Yes, the answer is B, and let me tell you how. So, because right now we already have learned V is equal to IR, and here because we have some polarization losses, and that we also have to account into that, and these losses are these things. And that's how we are subtracting it, right? So here, if we are saying first you have to see EC and EA. So EC is your electrode potential for the cathode and EA is your anode. So we have to subtract it so that we can have the voltage, right? But we have to minus the polarization losses, and this will be the loss due to activation, the loss due to resistivity, the loss due to concentration gradient, and the loss due to pH difference. So these are different losses that we have to subtract, and that's how the B answer is the right one. Okay, so the next question is, which of the following losses in microbial fuel cell is primarily caused by uneven depletion of reagents in electrolyte leading to concentration gradient near the electrode surface? And we have four options here: ohmic losses, activation losses, concentration losses, and pH difference losses. What do you think? What is the right answer for this? Okay, so let me see. Venkateshwara gave me D. Is this is like just read the question again. This is uneven depletion of reagent, and that will give you the concentration gradient. If we will have that concentration gradient, then this will give you some losses in the voltage, right? And if it is a concentration gradient, then what kind of loss it will give to you? Okay, so yes, so C is the right answer because here we do have concentration gradient at some places in the electrolytes. So there is the uneven depletion of the reagent. So some reagent are being consumed, and at some places the reagent are being not consumed. So there will be the difference in the concentration. So where there is more substrate is getting consumed, so it means this will be your diluted one. And the one where the substrate is not being utilized, then this will give you the concentrated. And we know that from dilute to the concentrated, so low concentration to the high concentration we will move, and this will give you some voltage difference, because this will not be according to what we want. This will be according, so your things or your electrons will be moved according to the concentration gradient, but not according to what we want. Okay, so this is how there will be some losses in the voltage, and that's how C answer will be the right one. So we do have different losses: ohmic losses due to resistivity, obviously. Then we have activation losses because of accumulation of gases at the interface between electrodes and electrolyte. Then we also have bacterial metabolic losses. We have concentration losses due to uneven depletion of reagent in the electrolyte, which causes concentration gradient in boundary layers and mass transfer through the membrane. And we also have voltage losses due to pH difference that we have in the cathodic and the anodic chamber. Okay, so these are different losses that we will have, and according to which we have to see how much electricity will be generated, because we have different losses in the voltage. Okay, so the next question is, which of the following strategy would most directly help increase power generation in microbial fuel cell? As in the last slide, we learned that there are some losses, and due to which the efficiency will be less than 100%, right? So, so to increase that efficiency or to increase the power generation, what strategy would help us the most? And we have four options here: increasing the anode area and reducing inter-electrode distance, using less efficient microorganism to slow down metabolism, avoiding catalyst to minimize electrode reaction, replacing oxygen with an unsuitable oxidant in the cathode. I think among that four options that I gave you, uh, one is odd one out, and that would be your right answer. Okay, so Dia gave me A answer. Let me see the answer. Okay, Venkateshwar also gave me A. So yes, so this is whenever we will increase the anode area. This is the place where microorganism to attach to that anode area, right? So whenever we are increasing the anode area, power generation will be more, and even reducing inter-electrode distance. So this is how we can increase the power generation. Using less efficient microorganism, no, we have to use efficient microorganism to increase the power generation. So this is not the right answer. Avoiding catalyst to minimize electrode reaction, no, we need more electrode reactions to generate the electricity, so we should have to have this catalyst. So this is not even the right answer. And replacing oxygen with unsuitable word is itself is giving you the wrong vibes, right? So that's how A answer is the right one. So along with that, we have different methods. So for example, if we add suitable metal ions using metal reducing microorganism, this is how we can also increase or improve the voltage power generation. This is the one way. The second way is we can use the doping of your catalyst on the electrode. Um, I I understand that you do know about doping, because this is 11th or 12th standard terminology. Then increasing anode area and decreasing inter-electrode distance, we can use more efficient microorganism and using more suitable oxidant. Uh, this is how we can increase or improve the voltage and power generation. Okay, so these are different methods or approaches, you can say. As I told you, we can have different type of microorganism in your bioelectrochemical cell, right? So you have to tell me microorganism and substrate pair that produce the highest current output in microbial fuel cell. And we do have four options: Shivanilla, putrefacium lactate, then geobacter, rhodophyrox with the glucose with porous graphite, and glucose with Woman graphite. So you need to tell me microorganism and the substrate that the organism needs, plus that would produce the highest current output. Okay, so I got D and C answer. Let me see what is the right one. So C is the right one. So we will use rhodophyrox, and substrate will be glucose with porous graphite. Then we will have the highest current output. Let me show you the table. So this is the table. So we have substrate right here. We do have substrate, and we have the microorganism. And if we are talking about the highest current and power, then highest one is this 74 and 33. So the microorganism we are using rhodophyrox, and substrate we are having glucose, and anode we have is the porous graphite. So from this combination, we will have highest current and power. So is this one? So this one C1 is, that's how is the right answer, right? Okay, so this was all about your secondary treatment. We talked about bioelectrochemical cells, right? And this is how we completed our secondary treatment. Now we are moving to tertiary treatment, and in which we have three ways. By adsorptive filtration, we will talk about this. Then we have the membrane processes, and we also have advanced oxidation processes. We will talk about these three methods by which we can remove some impurities, bacteria, viruses that we may have in the water, and this is how we can treat our water. So I have MCQ question now based on your tertiary treatment, and the question will start from adsorptive filtration. So, okay, so this is how we started. We started with primary treatment. We had debris screen, then grit removal, primary clarifier. Then we talked about this secondary. And here we talked about microorganism or activated sludge processes. We talked about it. Then right now we are on this tertiary. And here mostly we will talk about some filters, and for example that adsorptive filter, and even ion exchange, if you have heard, because in right now in most of that treatment tank that you have, or treatment body that you have with the RO, we will have the ion exchange also. So we will talk about adsorptive and ion exchange within that filter. Then we will use some disinfectant, and this is how we will purify the water, and this water will be discharged to the surface water. So right now we will talk about this one. Okay, so the question is, in tertiary water treatment, why is sodium bisulfite added after chlorination? To increase dissolved oxygen in water, to remove excess chlorine before discharge, to enhance coagulation of solid, to kill remaining bacteria and viruses. Why we are adding sodium bisulfite after chlorination? Dia gave me B answer. Okay, so Venkateshwar also. Yes, so B is the right answer. That's if we are adding sodium bisulfite, so this will remove excess chlorine before discharge, because chlorine will give you different type of smell as well. And obviously we do have some residual chlorine, so we do have residual chlorine as well in the water body, so that is helpful for us. But if we do have excess of chlorine, then it may be harmful to the human being. So to remove excess chlorine before discharge, we have to add sodium bisulfite. So that's how B answer is the right one. So here is something that tertiary treatment is the final stage, and we do have multi-stage cleaning processes. And here we will remove inorganic compounds. In secondary treatment, we removed mostly organic compound, right? But in tertiary, we will remove inorganic compound, bacteria, viruses, parasites, and make the treated water safe to reuse, recycle, or release into the environment. Whenever needed, we may add alum to remove phosphorus particle from the water. And this is we have done. Then we need to add sodium bisulfate so that we can remove the excess chlorine. And important tertiary processes that we have are adsorption, reverse osmosis, and advanced oxidations, on which we will have some MCQs. Okay. So the question is, which of the following is not commonly used in tertiary wastewater treatment? Very simple question. Reverse osmosis, advanced oxidation, adsorption, activated sludge processes. Okay, so you both gave me right answer. That is D, because this D is the part of what level of wastewater treatment? Can you give me the answer? Activated sludge process. Is it the part of primary or secondary? Okay, yes. So yes, this is the part of your secondary wastewater treatment. So that's how this is not the part of your tertiary wastewater treatment, and D will be the right answer. Okay, so right now we are talking about adsorptive filtration. Adsorptive filtration in the wastewater treatment primarily works because molecule pass through the filter pores by size exclusion. Molecules are transferred from fluid bulk to solid surface. Solids are mechanically trapped between coarse and fine media layer. Water molecule chemically react with the filter media. Little tricky, but I think you can give me the answer. So what is the principle of adsorptive filtration? From that word only, adsorptive, you can guess something right one. Okay, so you both gave me right answer. So this is molecule are transferred from fluid bulk. So we have wastewater, and that wastewater have uh, molecules that we need to remove. Okay, and this will be so for example, if we do have charcoal, let's say, and if this if the wastewater will be spread over this activated charcoal, then there will be this molecule will transfer to this activated charcoal, and the clean water will be gone. So this is how this will work. Molecule pass through the filter pores by size exclusion. So obviously it will pass through, but the principle is uh, it has to attach to that solid surface, right? So this is how B answer will be the right one. So this is something that we have. Coarse media, we may have coal. Then fine media, we may have silica sand. Finest media, we will have granite, and spot we may have of the gravel. And we will have the wastewater. Then there will be transfer of this molecule from liquid to the solid surface, and this is how you will get the treated water. Okay. So the next question is, which sequence best describe the mechanism of adsorptive removal in wastewater treatment? And we have adsorption, transfer to surface, diffusion into adsorbent. diffusion into adsorbent, transfer to surface, adsorption, transfer to transport to surface, diffusion into adsorbent and adsorption, transport to surface, adsorption and diffusion into adsorbent. So using this adsorptive process, how we can remove the some solid things that we have in the wastewater. So you have to tell me the sequence of this adsorption. So let me see. Dia gave me a C answer. Okay. So the thing is that something will be adsorbed. Adsorb means uh, some solid molecule that we have, or some molecule that we have, that would attach to some surface. Okay, so this will be the adsorption. So this will be your last step, right? Adsorption will be your last step. So that's how among B and C only you will have some answer. Then first you have to have transport to the surface, then there will be diffusion into adsorbent, and then it will be adsorbed. So this is how it will move on. So the sequence will be C, not B, and these are not the right answer, A and D, because the last step will be the adsorption. So just see here. First, so let's say this is your activated charcoal, let's say, and these are the molecule. First they will transport, okay, and then what will happen, then there will be diffusion, and then there will be adsorption. And we can have different type of adsorption. It may be physical, and it may be chemisorption. So using the charges, there may be adsorption. Okay. Okay, so the next question is, efficiency of adsorption in wastewater treatment is mainly controlled by. So again, we have to see the efficiency as well. What kind of material, what kind of technique is giving you the best option for wastewater treatment, right? So you have to tell me the efficiency is controlled by what kind of factor. And we have here four options: only the concentration of pollutant, only the surface chemistry of adsorbent, both surface chemistry of adsorbent and solution chemistry of uh, adsorbate, and only the operating temperature. Very simple, I think. And Venkatesh, I think, already have given me the answer. So it is C, obviously. So this is something, whatever wastewater we have, in that we also have to see what kind of inorganic material we will have, and based on that inorganic material we have to specify what kind of adsorbent we need to use. Okay, so that's how both the adsorbent and the adsorbate, the chemistry of these both things will determine the efficiency of adsorption in wastewater treatment. So that's how C answer is the right one. Okay, so little technical, but you can tell which factor mainly decide how pH affects the adsorption process. Temperature, zero charge potential for adsorbent, contact time, amount of adsorbent used. Little trickier, but I think you can tell. Okay, so Dia gave me B answer, and yes, B answer is the right one. So because we want to see how pH will affect, so this we can see using the factor that is zero charge potential. So this charge will vary. This zero charge potential will vary whenever we will have different pH, right? And based on that we can check whether pH is affecting the adsorption process or not. So that's how B answer is the right one. Okay. So why does granule activated carbon show low arsenic removal efficiency at neutral pH? And we have four options here. GAC surface is predominantly negatively charged. GAC dissolve at neutral pH. Arsenic is not soluble at neutral pH. Neutral pH blocks adsorption pores. So as I told you, whenever we will have the adsorption process, most of the time we people are using GAC, that is granule activated carbon. But this activated carbon is not showing great efficiency for the arsenic removal in the wastewater. So, but why? Why at the neutral pH? Venkateshwara gave me A, that GAC surface is predominantly negative charge. Yes. So, yes, A is the right answer. And let me give you some explanation about this. So, GAC has surface charge that depend on the pH of the solution. So, if we do have neutral solution, so pH 7, surface of the GAC will mostly have the negative charge, and arsenic also have negative charge. So if we will have negative charge, that means a charge will repel to each other and the adsorption efficiency will be lower. So what we have to do is we have to change the pH. In that condition only we can increase the efficiency for the arsenic removal by GAC, because at neutral pH the surface of GAC has a negative charge. Okay, so the next question is, how can the arsenic removal efficiency of GAC at neutral pH be improved? How we can improve the efficiency for arsenic removal by GAC? And we have four options here. Increasing agitation speed only, using a higher water temperature, impregnating GAC with multivalent metal ions, and reducing adsorbent dose. Okay, so you both gave me C answer, and yes, I also agree with the C1. So if we want to increase the efficiency even at the neutral pH, so what we can do is, along with GAC, we have to add some multivalent metal ions, and this is how we can remove this arsenic with uh, efficiency, right? Higher efficiency. So that's how C answer is the right one. Okay. Now the thing is that we need to check the performance evaluation of adsorbent, right? And how we can check the performance? Then we have three parameters or techniques by which we can see the performance of adsorbent. First, we need to calculate the adsorption capacity, and based on that adsorption capacity we may know what is the performance of that particular adsorbent. Then the next is adsorption kinetics, we will talk about this. And then spent adsorbent regeneration, so how much adsorbent is getting regenerated, right? And so based on that we will assess the performance of adsorbent. And Venkateshwara already have given me answer before. Okay, Diya also have given me the answer. So this question is, let me tell you the question first. So this is, which statement correctly describes the Langmuir isotherm model in absorption studies? So this is the one model that we use to calculate the adsorption capacity, right? And you have to tell me what different assumption we will have in the Langmuir isotherm model. And because you already have given me the answer, then with no time wasting, I'm giving you the right answer. So yes, this is B. So in that model, so first, why we are using that model? So we are using that model to assess the performance of adsorbent, the first thing. The second thing is we will have some assumptions in here, and the assumption is we are assuming that it is monolayer adsorption on uniform adsorbent surface with identical sorption site. Then only this model will work. So that's how B is the right answer. So this is the slide that I have taken. Uh, so here we do have the Langmuir isotherm, and this is to determine the specific uptake by your absorbent. And this is the equation that we have used. Based on that equation, you can also solve some numerical question as well. So this is just I want to show you. Then along with this Langmuir isotherm, we also have Freundlich adsorption isotherm you can also read about. So these are different methods by which we can assess the performance of adsorbent. Okay. So the next question is, which of the following correctly describe the pseudo first order adsorption kinetic model? So the next, so from here, just see. So we can also calculate the adsorption kinetics to assess the performance of your adsorbent, and based on that only we have this question, and this question is, which of the following correctly describe the pseudo first order adsorption kinetic model? So we have four options here. Adsorption rate is proportional to the square of the number of unoccupied sites. Adsorption rate is proportional to the number of unoccupied sites. Adsorption rate depend only on intraparticle diffusion. Adsorption rate is independent of equilibrium adsorption capacity. So what do you think? What should be the right one? And if it is the first order, then it will be directly proportional, right? So let me see. Venkateshwara gave me B answer. So yes. So in that model, or that pseudo first order adsorption kinetic model, we are assuming that adsorption rate is proportional to the number of unoccupied site. So whenever we will have unoccupied site, then more adsorption will be taken place. Okay, so this is how B answer is the right one. Okay, so this is from where we can see. So we have some models, important model for this adsorption kinetics, and these model are pseudo first order model, pseudo second order model, and we also have some intraparticle diffusion model. For example, Weber and Morris model. Okay, so you just need to remember these, the name of these models. And in pseudo first order equation, we people are assuming that this is directly proportional, right? Okay, so this is done, this is also done, right? Now we will have some question on this spent adsorbent regeneration also. Based on that, we can evaluate the performance of adsorbent. Okay, Which of the following is major concern when regenerating spent adsorbent in wastewater treatment? And we have four options here. The adsorbent may lose color after regeneration. Residual material may be toxic and require proper disposal. Adsorption capacity always increase after regeneration. Spent adsorbent cannot chemically treated. So let me see. Okay, my answer. So Venkateshwara and Dia both gave me B answer. So this is residual material may be toxic and require proper diffusion disposal. So for example, if we have activated charcoal, right? If we have activated charcoal and some molecule has been attached on it, some molecule has been attached on it, so we do not have right now, let's say, unoccupied site. So either we have to diffuse, either we have to dispose this activated charcoal because it is already some molecules already has been as adsorbed on it. Otherwise we have to regenerate this. So whenever we will regenerate it, so these molecules we have to remove from activated charcoal, right? And these may be toxic in nature, and that's how it will be difficult for us to dispose that. So that's how this is residual material may be toxic and may require proper disposal. Adsorption capacity always increase by regeneration? No. At start, whatever will be the capacity, that capacity will be maintained, but it will not be more, right? Then adsorptions, spent adsorbent cannot be chemically treated. No, we can treat it chemically as well. So B is the right answer. And this color is the absurd statement. So just see here, management of spent adsorbent which is rich in pollutant concentration is important issue for the success of this process. And obviously we can regenerate our adsorbent using different chemical treatment. However, management of residual material is an issue if residual material is toxic in nature. And that's how, if these are toxic in nature, residual materials, then what we can do is we can do the solidification. And we can also have to follow the land filling and hazardous waste management procedure if we have the toxic residual material. Okay. Uh, we talked about GAC, right? GAC is the part of adsorbent, but in that adsorbent, we also have ion exchange, and this ion exchange is used for the hardness removal. And you have to tell me which process take place in the softening of water. And we have four options here. Calcium and magnesium ion are replaced by sodium ion from resin. Sodium ion in water are replaced by calcium and magnesium ions from resin. Hydrogen ion replace sodium ions in hard water, and calcium and magnesium ion are converted into insoluble precipitates by the resin. Let me see the answers. Okay, so you both gave me A answer. Then yes, I also agree with this. A is the right answer. So why A is the right answer? So this is something, the hardness is due to, mostly due to uh, multivalent ions, right? And these are calcium and magnesium. So hardness is mostly by the calcium and magnesium. Okay, if we want to remove that hardness, we should have some processes. Just like GAC, activated charcoal, we may have resins. Okay, and that resins have Na attached to it. Okay, so sodium ion is attached to the resin. So whenever we will have resin that is attached with the sodium plus your wastewater, then what whenever we will have calcium and magnesium in the wastewater, so it will replace this sodium. So at last what we will get, calcium and magnesium with resin, right? And then this Na will react with something I will show you in the next slide. And this is how we will remove calcium and magnesium using exchanging the ions. What we are exchanging? We are exchanging sodium and magnesium with the sorry, calcium and magnesium with the sodium ion. So that's how this is ion exchange. Let me show you. So this is how we can do. So this is your resin. uh, this is your let's say resin RNA2, and in the wastewater we may have calcium and magnesium, and calcium and magnesium uh, maybe with chloride, sulfate, and carbonate. So it may be calcium carbonate, bicarbonate, calcium sulfate, calcium chloride. So likewise, these are the hardness that we have in the wastewater. So whenever we will have this with the resin, so what will happen? This will be attached with the resin, and then this will be in your water. Okay, and this is how we are removing calcium and magnesium from your water. But this calcium, sodium chloride, sodium sulfate, sodium hydrocarbonate, this will be in your water, and these are not that much harmful as as much as hardness calcium and magnesium have, right? So this is how we can use this ion exchange. But if we want to regenerate it, so what we have to do is, so this is the product at last we got, but we have to make the reaction with uh, sodium chloride, and this is how this magnesium and calcium will react with the chloride, will give you the magnesium and calcium chloride, but give you the original reagent again. So this is how we can regenerate it, right? So this reagent are available with sodium Na2. This is also available with hydrogen, and this is also available with hydrogen. And this is how we can whatever uh, compound we want to remove, based on that we will have very specific resin, and that specific resin will replace or exchange the ion with the one that is not wanted, right? This is unwanted. So based on that we will exchange the ion, and again we may regenerate it. So for example, if we want to regenerate this one, then we have to use hydrogen chloride. If we want to regenerate this resin, then we have to react it with the sodium hydroxide, and this is how we will get the original resin. So this is the principle of ion exchange. And in along with ROs, mostly right now in the water purifier, we do have ion exchange technique within that air purifier along with the ROs. Okay, so in the tertiary treatment, we also have water disinfectant, right? So we have disinfectant, for example, chlorine, and you have to tell me which method, which statement about water disinfectant method is correct. And we have four options here. Chlorination leave no residual disinfectant in water. Ozonation is effective but does not provide long-term residual protection. UV light treatment add chemical that maintain residual disinfection. All three methods provide the same type of residual protection. Okay, so Dia and Venkatesha both gave me a B answer. Then yes, B answer is the right one. Chlorination, whenever we are using chlorination, so the advantage of chlorination is that some of the chlorine molecule will be within your treatment water. So whenever there will be some bacteria that will go into that water, so that chlorine can disinfect your water. So for the long term, if we want water for the long term, it is suitable that we use the chlorine, because that have the residual capacity as well at later stage, because chlorine will be present in your water, and that's how it can disinfect your water for the long term. So this leaves no residual is the wrong statement. Ozonation is effective, yes. Ozonation is the one of the advanced disinfectant method, but it would not provide long term residual protection, because you know that ozone and ozone is very short term, and it will be break down into water, O2 and O, and this breakdown, and this is very short term species that we will have after ozone breakdown, right? And that's how this is not for the long term. UV light treatment add chemical? No, UV light treatment doesn't add chemical. So this is again the wrong statement. And three method provides same type of residual protection? No, because chlorination give you residual protection, but ozonation and UV light treatment doesn't provide you the uh, residual disinfectant, right? So that's how B answer is the right one. Okay, so we talked about adsorption, right? We talked about uh, adsorption in your tertiary treatment, right? Now the next topic is membrane processes. So we can use membranes. The very simple example is that we people sometimes use cotton cloths to purify the water, right? So this is something is known as the membrane process. So we do have membrane process. What different type of membrane we can use? So membrane is something, some filter like this, right? So, and what different type of membrane we will have? Different size separation. Then we have organic and inorganic membranes. Then we will talk about the principle of working of this membrane process. Then shape and modules of different membrane, how the water will be transport in membrane, and liquid diffusion through pores. So right now we will have question on membrane processes. The another example of membrane processes is in cell. So if we do have either human cell or plant cell or animal cell, then these the cell wall or your plasma membrane outer of this cell is very selective. What it need water or not into it, if it need sodium or not. That means there is a particular type of membrane that is allowing some chemical to enter in and some chemical to exit out, right? So this is very simplest example of membrane, and this membrane we can also utilize to purify the water. So okay, coming to the membrane. So just read about this. Membrane processes are physical diffusion process. Just see, it is a diffusion process of the particle in membrane. So for example, some of your particles, some of your molecule will enter into it. So there will be diffusion, but some will be resisted. So they function because certain type of membrane allow particle with particular characteristics only to pass through them. And based on what type of material we are using in that membrane, we will have the permeability of particular type of particle. Then membranes are thin and porous sheet of material able to separate contaminants from water when a driving force is applied. This is also important. We have to give, so for example, here we are giving the pressure. So this is your driving force. We also have, like, we should have some driving force. It may be pressure, it may be the concentration gradient. So the simplest example is resin. So whenever we are soaking it into water, then it will be swallow down, and this is just because the concentration difference, right? So because that resin also have some membrane outer to it. Okay, so the question is, which of the following correctly matches the membrane type with its driving force? Reverse osmosis, electric field, electrodialysis, pressure gradient, nanofiltration, pressure concentration gradient, membrane distillation, ion exchange. Membrane type, first you have to tell, and then uh, that will match correctly to the driving force. What do you think? What type of membrane will have what type of driving force that will give you the purification of water? Any guess? Okay, so it is nanofiltration, and in that nanofiltration we may have either pressure or even concentration or both gradient that we can maintain uh, to filter even the nanoparticle from your waste water or even from your very normal water, but it may be harmful to drink. So we can use the nanofiltration, and let me show you different. Okay, I think next slide have that explanation. Anyway, so which membrane process is best suited for producing nearly pure water with most solute removed? Microfiltration, ultrafiltration, reverse osmosis, electrodialysis. Which is best suited for producing nearly pure water, and that would remove mostly all the solutes. Not ultra, not ultra. Okay, so, let me show you. So this is reverse osmosis. C is the right answer. Let me show you this one. So this is something, you have water, and water may have bacteria, viruses, and some solute that is dissolved into your raw water. Then if we do have some membrane, and that membrane have the pore size of 10 to 0.1 micron. Okay, so that will come under microfiltration. So that big would be removed, but some of that would cross that membrane. Then we will have the ultrafiltration, where we have the filter, the size of that filter pore will be 0.1 to 0.01, and this is how some of the molecules will pass on only. Then we have the nanofiltration. But if we are talking about the reverse osmosis, RO, then here we have 0.001 to 0.0001 micron size. So it means most of your particle, viruses, bacteria will be resisted by this membrane, that is the reverse osmosis, right? And the thing is, that's how, why we are saying that uh, whenever your water uh, doesn't have much problem, then why we should not use this reverse osmosis, or we should not use the ROs. The problem with the RO is that it may be that some of the molecule, let's say this molecule may be useful for us, or let's say we have some iron, or let's say some manganese that we need in the body, and these are known as the trace elements. Let's say we need some trace elements in our body, and that trace element may found in the groundwater. Okay, and if we are using ROs, that it means this trace element that are beneficial for our body will also be removed under this process. And that's how sometime we say that whenever your water does not have much problem, then you should not use the ROs, because some of the important trace element that we need for our body are getting removed using that technique, right? But if you have literally bad water that is coming at your home, then obviously we should use RO, right? So this is the most suited based on the pore size, right? Based on the removal of your particles. Okay. So the next question is, in wastewater treatment, a key advantage of membrane process is what? They always prevent membrane falling. They require no driving force for separation. They can achieve disinfection without chemical addition. They never produce residual polluted water. What do you think? What is the key advantage of membrane processes? Okay, so Venkatesh gave me a D answer, but the answer is C, because they can achieve disinfectant. Disinfectant is used to kill the viruses, bacteria, right, that we have. And this can be achieved by the membrane processes using RO, even without adding any chemical addition, or without adding any chemical. So that's how membrane process is also doing the work of disinfectant also. So that's how this is the key advantage. They never produce residual uh, polluted water. No, this is exactly not the right answer. So that's how C will be the right one. So these are the advantages. So we will have high performance, uh, compact unit. So we will have very less space that is needed, and if within that less space we can purify the water. Very simple operation, and membrane available can be used to separate many kind of contaminants, and disinfection can be performed without chemicals. So these are the advantages of the membrane processes, but we also have some disadvantage. For example, because membrane is trapping the particles, right, or trapping the bacteria, viruses, so it can give you some falling smell, production of polluted water from backwashing. Obviously, whenever your membrane will be filled with particle, then you have to use backwashing to clean that membrane. So here you will have the production of polluted water. Then membrane have to be replaced on the regular basis, then only we can have some advantages of membrane process. Okay. So coming to the next question, and this is, which membrane process is correctly paired with its driving force? And we have electrodialysis, pressure gradient, reverse osmosis, steam pressure, membrane distillation, temperature difference, microfiltration, electric field. Which membrane process is correctly paired with its driving force? Any guess? Microfiltration, electric field, no. Microfiltration is mostly based on your pressure gradient or concentration gradient. Okay, so give me right answer. So C is the right answer. I do have table, I show you. So uh, in the filtration, microfiltration, ultrafiltration, and nanofiltration, the driving force is pressure. For the reverse osmosis, the driving force is pressure as well as the concentration. For electrodialysis or reversal electrodialysis, the driving force is electric field. But if we are having the membrane distillation, the division factor is temperature and it is steam pressure, right? Okay, so coming to the next question, as we already have discussed about this, which of the following correctly represent the pore size range for ultrafiltration membrane? I already have shown you a figure, and if you remember, then you can give me the answer. The pore size range for ultrafiltration membrane. Dia gave me B answer. Okay, so yes, B answer is the right one. And these are the size that we have. So for the microfiltration, for ultrafiltration, and for the reverse osmosis, the pore size in angstrom and micron, it is given. So most of the time we people use in microns. And what material we use, so for example, for microfiltration we mostly use cellulose acetate, poly sulfone ceramics, and in ultrafiltration also we use the same, but in reverse osmosis we can also use aromatic polyamides, right? And these are the pore size that we need. Then membrane pressure drop, as it will as the particle will move, so there will be always a pressure drop. Then permeate, so permeate is what we will get at last. So what will be the product? So initially we will have initially we have water with some bacteria, viruses, some dissolved molecule. So permeate is the end product that you will get after filtration. So in microfiltration you will have water plus dissolved molecule. In after ultrafiltration you will get water plus small molecule, and in reverse osmosis most of the solute will be removed and you will get just the water. And retentate, so this is what will be removed. So water and large suspended particle will be removed. In ultrafiltration, water plus large molecules will be removed, and reverse osmosis, water and some solute will be removed. And that's why there is always a pipe in the arrows, and because that is giving you some water that will be taken away, that is polluted water that will have a high concentration of solute in it, in that. Okay, so the next question is, which of the following membranes can withstand the highest temperature and widest pH range? So as I told you, different material, so different membrane is made up of different material, and you have to tell me which one can withstand the highest temperature with widest pH range. And we have four options here: cellulose, diacetate, polyamide, zirconium oxide, and aluminium oxide. Venkateshwara gave me B and Dia D. And yes, D is the right answer here. It is aluminium oxide. Let me show you the table. Uh, so first we have organic membrane as well as inorganic membrane, and we have to see the maximum temperature that it can withstand and the pH range. And if you see this one, aluminium oxide, if you see the aluminium oxide, then this can withstand the maximum temperature more than 900 degrees Celsius and pH from 1 to 14. So that's how this inorganic membrane made up of aluminium oxide is best suited for the where there is a high temperature and more pH range. Okay, so the next question is, in membrane separation process, the portion of feed that does not pass through the membrane is called. So we have membrane and water will cross this, and some solute will be here in that side only, and your pure water will be out. So here that does not pass through the membrane. This will be known as what? Let me see. So I got C answer. So yes, so retentate is retention. It came from retention. So which is retained, which are not passing out, right? So this will be known as the retentate. So we have some terminologies here. So we have feed mixture. Feed mixture is your wastewater that have a lot of solute. Then we have membrane, and we can also have sweep. So I have one MCQ question on this sweep as well. Then what we will get is the permeate, and what will be rejected one will be known as the retentate, right? Okay. In general membrane processes, a sweep refers to a cleaning step used for fault membrane, a liquid or gas that helps remove the permeate, the concentrated portion of the feed, the polymer coating on the membrane. Okay, so I got B from both of you. So yes, so this is any gas or liquid that will remove the permeate. So this will push. That's how this is a sweep up. So B is the right answer. Okay. So the next question is, which of the following is not a common membrane geometry? Now we are talking about the shape of your membrane, right? And we have four different type of shape that I gave you: flat, asymmetric, or thin film composite, tubular, hollow fiber, and cubic. Let me see the answer. Okay, so you both gave me D answer. So yes, D answer is the right because this is not common membrane geometry. Let me show you the figure. So these are important membrane geometry are flat, asymmetric, or thin film composite. It may be tubular, it may be hollow fiber, or it may be monolithic. And you can see here, if it is a flat, then this is just the flat, just like a tile, or if it is a tubular, then this is just like pipe. And then hollow fiber, this is again like pipe only, but like little hollow, more hollowness into that. And we may have monolithic, so this is honeycomb like structure we may find within that membrane. So these are different geometry or shapes that we people use to develop this kind of membranes. Okay, so the next question is, a spiral wound module is generally associated with which type of membrane geometry? And we have four options here: tubular, flat sheet, hollow fiber, monolithic. Spiral wound module. So Dia gave me B answer. Okay, so yes, this is, yes, so this is flat sheet. So first you have to see what do you mean by this membrane module. So this membrane module is a complete unit that that will have membrane, that will also have a housing, even the feed inlet, the concentrate outlet, and permeate outlet. So this is like a system in which we will have a lot of things, right? And we do have four major type of this module, and this module can be plate and frame module. This can be spiral wound module, and multitubular membrane, and the hollow fiber membrane. So these are different type of membrane module we can have. So here you can see, if we have a plate and frame, then large tube and hollow fine fiber. So here you can see we have feed water, we have product water, we have permeate. So this is kind of complete unit, which is known as uh, uh, which is known as your module, membrane module, right? Okay. Which membrane module has the highest packing density but poor falling resistance and poor cleaning ability? And we have four options again: plate and frame, spiral wound, tubular, and hollow fiber, that have the highest packing density but poor falling resistance and poor cleaning ability. Venkateshwara gave me a B answer. Okay. So, but it is hollow fiber. Let me show you this one. So, different type of membrane module we have, and along with that, this table is giving you the packing density, resistance of falling, ease of cleaning, relative cost, and main applications. And if you see at the hollow fiber, then this is giving you the poor resistance of falling, poor ease of cleaning, and low relative cost. But packing density is very high, that is 500 to 9000 meter square upon meter cube. So that's how this is giving you higher density, but poor resistance of falling and poor ease of cleaning. So that's how that D answer is the right one. Okay, so the next question is, which membrane module allows the possibility to analyze permeate at each stage but has the drawback of low packing density? Again, the question is on membrane module, and this is like the advantage is it is giving you the possibility that we can analyze the permeate at each step, so what we are getting output, but the drawback is it has the low packing density. Okay, so Dia gave me a answer, that is plate and frame. So yes, so the answer is plate and frame. So just see here again we have one table, and that one table have the module geometry, advantage and disadvantage. So for plate and frame, there is the possibility that we can analyze whatever we are getting at each stage, but the drawback is it has low packing density. For the spiral wound, we do have high package density and low cost, but cleaning is very difficult for this, and pre-treatment is compulsory because the cleaning in place is very difficult. For the tubular, advantage is limited pre-treatment. We do not need to have this pre-treatment and well adapted to cleaning in place. But again, the drawback is it has low packing density and it is taking a lot of energy for the process. For the hollow fiber, it has very high packing density, low energy consumption, and low cost. But the thing is, a pre-treatment compulsory uh, for in-out mode. So these are the advantage and disadvantage of your different module geometry. Anyway, so coming to the next question. In membrane transport, which mechanism typically occur in non-porous membrane instead of porous one? So till now we were talking about porous that have some pores, and that pore will not allow different particles to go through. Okay, so but we also have some mechanism that typical occur in non-porous membrane. And so can you tell me that mechanism that typically occurs in non-porous membrane? And we have four options here. Movement of solute with the solvent due to pressure gradient, migration of molecule along concentration gradient inside membrane pores, restricted movement of large molecule through narrow pores, molecular dissolution into membrane followed by diffusion across it. What do you think? What is the right answer? Which mechanism typically occur in non-porous membranes? So, two processes, dissolution and diffusion. Dissolution and diffusion. Venkateshwara, dissolution and diffusion are the two mechanisms that can also happen in non-porous membrane. So, can you tell me now the right answer? Okay, so D answer is the right one. So just see here, the mechanism of transport in membrane. So we may have bulk flow through the pores. We may have diffusion through the pores. We may have restricted diffusion through pores. But if we do have solution diffusion, that can be occur through dense membrane. So that's how that D answer is the right one. Is it clear? Why A is not the right, but B is right? So, okay, coming to the next one, the last but not the least. So this is advanced oxidation processes. So what will happen? Whatever we have, molecules that we have in the water, so we do have some oxidant. That oxidant will react with that molecule, and this is how it will give you CO2 and something like this. And this is how we will have the purified water. So the working principle is we will use some oxidant, right? And the very dangerous and very strong oxidant that we have is hydroxyl radical, OH, and that we will use in different AOP, that is advanced oxidation process. But this AOP uh, oxidation process can be light driven, can be can be done by benzene oxidation, and can have some advantage, disadvantage, and we do have different prospect about this advanced oxidation process. So I think it will take 10 minutes to cover, and then we will have like dinner time. Okay, so this is the last topic. So we have advanced oxidation process, that is AOP, primarily rely on which species for degrading contaminants in wastewater? Dia is very quick in giving me answer B. So this is hydroxyl radical. Yes, Venkateshwara also gave me the same. So yes, So most of the time we people are using hydroxyl radical because this is very strong oxidant by which we can degrade the contaminants whatever we have in the wastewater. So this is hydroxyl radical. So okay, so the thing is, why we are using this hydroxyl radical only? Why we are not using another different things? So the thing is, it is highly reactive. So whatever compound we have in the water, that can be degraded very fast using hydroxyl radical. It is not selective. It means for a lot of, for a variety, for a thousand of contaminants, hydroxyl radical can be used for the degradation. It is easily generated. It is very powerful oxidant. It is short-lived and harmless. This is again the advantage, that this is short-lived and it is harmless and control the rate of reaction. So these are the positive side of why we are using hydroxyl radical for the advanced oxidation process to treat the water. Okay, so based on that figure, you can tell hydroxyl radical used in AOP are best described as long-lived and highly selective, short-lived, highly reactive and non-selective oxidant, stable molecule that require catalyst for the reaction, weak oxidant that target only inorganic pollutant. Uh, yes, B is the right answer. You both gave me B, and we already have learned this is short-lived, this is very reactive, and it is not selective in nature, right? This is non-selective, and it can target a lot of inorganic and organic pollutant. Okay, so that's how it is. It is very strong technique by which we can remove the unwanted contaminants. Okay, so the thing is, we can generate this hydroxy radical using a different compound. So for example, uh, okay, so you have to give me the answer, and they already have given me. So which of the following is not typically used in advanced oxidation process for generating hydroxyl radical? So how we can generate this hydroxyl radical? So we have four options: ozone, hydrogen peroxide, a UV light, and activated carbon. So obviously D is very absurd option, and that's how you people are very quick in giving me an answer. But anyway, ozone, hydrogen peroxide, and UV rays are used to generate the hydroxyl radical, and D is the like not the way by which we can generate the hydroxyl radicals, and then D is the right answer. Okay. So arrange the following oxidant in decreasing order of oxidation potential. So which oxidant have very strong oxidation potential, and which oxidant have like low oxidation potential? And you have four oxidant here: hydroxyl radical, atomic oxygen, ozone, hydrogen peroxide. And you have to give me the sequence for this. So the thing is very clear that hydroxy radical is the strongest one, right? So based on that, I think B and among B and D will be only the right answer. Now you have to tell me either it is B or it is D. Okay, so one gave me B and another gave me D, and let me show you. So answer is B. So yes, so the lowest oxidation potential is in hydrogen peroxide. Then more will be to the ozone, more to the atomic oxygen, and strongest with the hydroxyl radical. So B is the right one. And this is the value oxidation potential is given. So it is highest for the hydroxyl radical, then atomic oxygen, ozone, hydrogen peroxide and HOCl. Okay, which of the following correctly represent the three essential steps of advanced oxidation process? Adsorption, hydrolysis, sedimentation, formation of hydroxyl radical, attack on pollutant, mineralization, coagulation, flocculation, filtration, aeration, oxidation, precipitation. Which one is the right answer? So Dia gave me B answer. Yes. So B answer is the right one. Because adsorption, hydrolysis, sedimentation, adsorption is the part of your adsorption process, but not of the AOP. Coagulation and flocculation, it was the part of your primary treatment. And this is not about the secondary treatment. Then aeration, oxidation, and precipitation, this is also the part of your primary treatment, and this is not the part of tertiary, and within that tertiary it is not the part of advanced oxidation process. So within that AOP, first we will generate hydroxyl radical, then we will have some reaction among this hydroxyl radical and the pollutant, and then this pollutant will be mineralized, and this is how all the uh, contaminants will be removed. Okay, as I told you, we can generate hydroxyl radical using UV rays and also using hydrogen peroxide. So this is UV hydrogen peroxide process. So hydroxyl radical are formed mainly due to breaking of HO bond in water molecule, homolytic cleavage of OO bond in hydrogen peroxide, reaction between UV light and dissolved oxygen, oxidation of chlorine compound. So Venkateshwara gave me B, and yes, the answer will be B only. Why this is B? Because we have hydrogen peroxide. Then we will give the ultraviolet radiation to the hydrogen peroxide, because ultraviolet radiation have very high intensity, because it has a very short wavelength. That's how this has a very high intensity. So that ultraviolet radiation will break that bond of this OO among hydrogen peroxide, and this is how it will give you the hydroxyl radical. So this is answer will be B, homolytic cleavage of the OO bond in hydrogen peroxide. So this is how it is look like. H2O2, then ultraviolet radiation we will give, and this is how it will give you the 2 hydroxyl radical. Okay. Okay, so which additional radical is produced along with the hydroxyl radical during the UVHOCl process? So in place of hydrogen peroxide, if we are using HOCl to produce the hydroxyl radical, then what additional radical will be produced along with that? So we have peroxide radical, chlorine radical, nitric oxide radical, and the hydrogen radical. Obviously, if it is HOCl, then chlorine will go somewhere, right? So this will give you the chlorine radical. Yes. So B answer will be the right one. So here you can see this will give you the OH and the Cl radical. Okay, so we can also produce hydroxy radical using TiO2, that is titanium titanium oxide. What happens when TiO2 surface is irradiated with UV light? Only heat is generated. Electrons are excited, producing an electron and a hole. Titanium oxide dissolved in water, only hydrogen gas is released. What do you think? Yes, so both of you gave me B answer. So yes, so whenever there will be the UV rays on titanium oxide, then electron will be excited, and this will give you the electron and the hole. Okay, so we also have light-based wastewater treatment method, and that would include what? You have A, B, C, D option. So UV oxidant, UV ozone, photophanton and photocatalyst and chlorination. Okay, so from that, like among these options, you can see if it is a light base, then we have to give some light, and that light should have high intensity. So that's how we are using UV oxidant, UV ozone. Photo means light again, photophanton, and photo means light, photocatalysis. Okay, but this chlorine, ozonation, coagulation, sedimentation, these are not based on the light, right? Micro, ultra, nano, these are the part of membrane process. Ion exchange, adsorption, flocculation, precipitation, some are based on some chemical and some are based on the adsorption. But A option is giving you the light based wastewater treatment method. So A answer will be the right one. So I think I have one or two questions right now left. So the during the oxidation of benzene by hydroxyl radical, which is the final outcome if sufficient hydroxyl radical are present. So if we are generating hydroxyl radical by oxidation of benzene, then along with hydroxyl radical, what will be the final output? So this will be complete mineralization into water and CO2, because this is benzene, right? So benzene have CHO. So there will be complete mineralization of this CHO into water and carbon dioxide. Okay, so I think these are the last question, or the second last. Advantage of AOP. They transfer pollutant to another phase for disposal. Hydroxyl radicals selectively react with only one type of contaminant. They can remove multiple organic contaminants simultaneously without forming new hazardous substances. Heavy metal cannot be removed by AOPs. Yes, because we are talking here about advantages, then this will be they can remove multiple organic contaminants simultaneously without forming new hazardous substances. But what is the major disadvantage of AOP? They produce water as a final product. They require continuous input of chemical reagent, making them costly. They can remove a wide range of pollutant. They achieve disinfectant in some design. Because it is disadvantage, then I think this will be very easier for you to give me the answer. Yes, so yes, B is the right answer, because we have to give some chemical reagent, then only hydrogen peroxide will generate. And if you know that this is very short term, then it means we have to give the continuous input of the reagent, then only hydroxyl a radical will be generated time to time whenever we need it, right? So that's how B is the right answer. Recent research trend in AOP include, this is we are developing modify AOP, because like titanium oxide, and we are doping it with the non-metallic elements or ultrasonic assistant AOP, so that we can improve the efficiency. So this is the recent research trend that is going on in AOPs. So this is, I think, done. So thank you so much. Thank you, Venkateshwar. Thank you, Diya, for being here patiently for the 2 hours. And thank you so much. And now this is the time that you can go and you can have dinner. We will meet next Monday from 7 to 9. And thank you so much. Thank you so much, everyone. Now